Mesothelioma is an aggressive form of cancer that develops from the mesothelium, a protective lining that covers many of the internal organs of the body. The mesothelium is found lining the lungs, the pleura, abdomen, the peritoneum, heart, the pericardium, and testes. The vast majority of mesothelioma cases are caused by exposure to asbestos in the workplace or environment. Developing an effective vaccine for mesothelioma faces significant scientific challenges. The cancer arises due to asbestos exposure often decades earlier, which means the immune system has had a long time to develop tolerance to tumor antigens associated with the initial asbestos injury. By the time cancer develops, the tumor may have evaded immune detection and generated an immunosuppressive microenvironment that prevents the immune system from attacking the tumor cells. Some of the key challenges in mesothelioma vaccine development include identifying tumor-specific antigens. Mesothelioma cells lack definitive tumor-specific antigens that can be targeted to provoke an antitumor immune response. Most tumor antigens in mesothelioma are also expressed on normal cells of mesothelial origin or other cell types. This lack of tumor specificity means a vaccine may not be able to precisely distinguish malignant from healthy cells, potentially limiting its effectiveness or resulting in autoimmune off-target effects. Extensive research is still needed to define novel antigens uniquely expressed or overexpressed in mesothelioma tumors versus normal tissues. Tumor heterogeneity. Mesothelioma tumors are known to be quite heterogeneous, containing different subclones of tumor cells with varied antigen expression profiles and molecular characteristics. This heterogeneity contributes to tumor evolution and enables cells to evade detection and targeting by the immune system. A single antigen vaccine may not be sufficiently broad to induce immune responses against the array of antigen variants expressed across a heterogeneous tumor. Multivalent vaccines targeting multiple antigens may be needed but are more difficult to design and develop. Immune suppression in tumor microenvironment. By the time mesothelioma is diagnosed, the tumor has already established local and systemic mechanisms to suppress antitumor immune responses. Factors secreted by tumor and immune cells in the microenvironment, such as TGF-β, IL-10 and PD-L1, actively paralyze immune effector cells. This immunosuppressive milieu prevents effective trafficking and infiltration of vaccine-activated T cells to eliminate tumor deposits. Overcoming established immune suppression poses a formidable hurdle for vaccine efficacy. Challenges of mesothelioma disease stage. Patients with newly diagnosed, early-stage mesothelioma may be the best candidates for vaccine-based therapy. However, mesothelioma is usually detected at advanced, metastatic stages when massive tumor burdens have accumulated. At later stages, systemic dissemination and immunosuppression make vaccine responses and tumor control much more difficult to achieve. Improving early detection strategies could help address this challenge by identifying candidates at earlier, more amenable disease stages. Latency period. After asbestos exposure, mesothelioma has a remarkably long latency period, sometimes 40 to 50 years, between initial asbestos exposure and clinical cancer development. During this extended latency phase, the immune system is continuously tolerized to exposure-induced damage in transformed cells. By the time tumors emerge, both central and peripheral tolerance may have rendered the immune system unable to recognize and react against tumor cells deriving from the initial rare malignant transformation. Reversing this state of immunosuppression and ignorance is extremely difficult. Lack of good animal models. Effective preclinical models to test mesothelioma vaccines are challenging to establish. Commonly used animal species do not reliably develop spontaneous mesothelioma with asbestos exposure like occurs in humans. Xenograft models using human mesothelioma cell lines fail to recapitulate key aspects of tumor immune cell interactions, long latency period, and immunosuppressive microenvironment seen in patients. The lack of physiologically relevant animal models is a barrier for proper preclinical evaluation of vaccine efficacy and mechanisms of action or failure prior to human testing. Financial and regulatory hurdles. Developing an effective therapeutic cancer vaccine involves major financial investments considering the scientific and clinical challenges. High costs are incurred in target discovery, preclinical testing, manufacturing, Phase A3 clinical trials, and post-marketing requirements. For rare cancers like mesothelioma with small patient populations, gaining regulatory approval and market viability can be difficult due to the prohibitive costs of drug development. Public and private funding sources are limited for vaccines compared to other cancer drug classes. This impediment hinders progress in the field. Designing and validating an effective therapeutic vaccine against mesothelioma faces considerable scientific and practical barriers. Major challenges involve identifying suitable tumor-specific antigens, overcoming tumor heterogeneity and established immune suppression, properly timing vaccination to amenable disease stages, and reversing long-standing immune tolerance from asbestos exposure decades prior. Improving understanding of mesothelioma immunobiology, establishing relevant preclinical test systems, addressing financial limitations, and expanding patient cohorts early in the disease course can help advance efforts to develop a mesothelioma vaccine. But considerable further research and collaboration across disciplines will still be needed to overcome these formidable challenges.